It's kind of wild to think that, like, even today, we are still fighting mosquito-borne diseases
Yeah.
like malaria, dengue, Zika.
Right.
These aren't, problems that are just, like, far off in some other country. They affect
Mhm.
millions of people around the world.
Yeah.
And we have things like insecticides and bed nets, but those solutions are
Right.
expensive and difficult to maintain long term.
Yeah.
So today we are going to be diving into something
Okay.
that has the potential to be revolutionary, and that's GeneDrive technology.
Cool.
Think of it as a genetic tool that can reshape how we control diseases.
Mm.
And it could even impact species beyond just mosquitoes.
That's really interesting.
What's fascinating about gene drives is that they could rewrite the genetic makeup of entire populations.
That's right.
So we're not talking about just managing mosquitoes here.
Mm-hmm.
We're talking about eliminating their ability to transmit diseases.
Exactly.
And that's a huge shift from what we're doing today.
Yeah.
So we have a ton of research to get through today. A lot of it is about this software called MGDrivE,
Okay.
which is essentially a simulator for gene drives.
Gotcha.
And it helps scientists figure out, how these changes would play out in the real world.
Yeah, that's important.
Yeah, for
Yeah.
sure. Before you release something this powerful, we need to understand how it's going to work.
Exactly.
And MGDrivE lets us, basically run these scenarios virtually,
Mm.
looking at things like the life cycle of the mosquito,
Okay.
the spread of diseases,
Right.
and even the genetic makeup of that specific population you're targeting. It's like a dress rehearsal for gene drives.
I like that.
And there are actually multiple versions of this MGDrivE software.
Really.
There's MGDrivE 1, 2, and 3.
Wow.
Each version adds new layers of,
Right.
complexity and insight.
Okay.
So we'll be unraveling all of that today.
Cool.
But before we get too far ahead of ourselves, let's start with the basics.
Sure.
What are gene drives,
Sure.
and how do they actually work?
So imagine you have this gene that you want to spread throughout an entire population.
Okay,
Usually inheritance is like a coin toss,
50-50.
right? 50-50 chance of passing it on.
Right.
Gene drives use a technology called CRISPR to basically guarantee
Okay.
that the desired gene is almost always inherited.
Oh, wow.
It's like rigging the coin flip to always land on heads.
Wow, so CRISPR is the key here.
It is.
We hear about it all the time, but as a refresher,
Yeah.
how does CRISPR actually make gene drives possible?
Well, CRISPR is like a pair of molecular scissors
Okay.
that can cut DNA at a precise location.
Mm-hmm.
And scientists use these scissors to insert the gene drive alongside the desired gene
Okay.
into the target organism's DNA.
Okay.
When that organism reproduces,
Yeah,
the gene drive copies itself onto the other chromosome.
gotcha.
And that ensures that it gets passed down to almost all of the offspring.
So it's not just passing on the gene, it's passing on the ability to make sure
Right.
that gene is inherited, which then creates this chain reaction through the population.
Precisely.
Oh, wow.
And that's fundamentally different than, say, using insecticides,
Right,
which have to be reapplied all the time.
right.
A successful gene drive could, in theory, create a lasting change with just a single intervention.
Okay, so this is where things get really interesting because we're not just talking about
Right.
one type of gene drive.
Yeah.
There's something called population suppression,
Okay.
which aims to reduce the number of mosquitoes.
Mm-hmm.
And then there's population replacement, which focuses on changing the mosquitoes so they can't even transmit disease.
You got it.
And to make sure we're making informed decisions, we use MGDrivE
Yeah.
to model all of this. And MGDrivE E2 specifically
Okay.
is a huge step forward
In what way?
because it incorporates so much detail about the environment where these mosquitoes live.
Oh, interesting.
It's not just some generic mosquito simulation.
Oh, yeah.
We're talking about taking into account things like rainy seasons versus dry seasons. Oh, wow. Which obviously affects how many mosquitoes are around
Right.
for the gene drive to spread through.
For sure.
And MGDrivE E2 even simulates the transmission cycle of the disease.
Wow.
So it's following how it moves
Right.
between mosquitoes and humans.
Gotcha.
It's like a virtual world where researchers can watch a gene drive play out
That's amazing.
in a complex ecosystem.
Yeah.
Now, this might sound super technical, but MGDrivE E2 uses
Okay.
something called stochastic Petri nets.
I've heard of that.
What does that even mean?
It's basically just a fancy way of saying they build
Okay.
a very adaptable and flexible system for modeling.
Oh, okay.
Think of it like a flow chart that allows researchers to account for
Okay.
all sorts of variables and possibilities.
Gotcha. So we've got this really detailed simulator.
Yeah.
But it gets even cooler because MGDrivE E2 also allows researchers to model
Uh-huh.
these like really cutting-edge gene drive designs.
Oh, cool.
Like, for example, there's this idea of using small molecules, almost like chemical switches,
Oh,
that
wow.
control when and where a gene drive is active.
So you could have a gene drive that's only activated in a certain area
Yeah.
or during a particular season.
Yeah, exactly.
That's really precise.
Yeah, small molecules can give us that kind of control,
Yeah.
which makes gene drives more targeted and predictable.
Yeah, for sure.
It's like a gene drive with an on-off switch.
I like that.
And to add to that, they're also exploring self-eliminating gene drives,
Oh, cool.
which is like having a built-in expiration date,
Right?
which is reassuring given some of the concerns about unintended consequences.
Exactly.
What if we release a gene drive
Yeah.
and then later we discover some unintended side effects?
Right.
Well, with the self-eliminating drive, you can program it to basically fade out after a certain amount of time,
Oh, wow.
leaving the original population largely untouched.
So it's not a one-way ticket.
Right. It's like a
Wow.
round trip for the gene drive.
I like that.
And this allows us to, explore gene drives and their potential without the risk of like irreversible change.
Yeah, for sure.
Now I know what you're thinking.
What am I thinking?
This all sounds amazing,
Yeah, it does.
but why should I care
Yeah, why should I care?
about all these self-destructing gene drives and these chemical switches? Well, it's all about safety and control.
Gotcha.
These advancements give us the tools to make gene drives
Yeah.
much more precise, predictable, and less risky.
Right.
And that's super important if we're going to be using them responsibly.
I agree.
And to illustrate this, there's a really cool figure in one of the papers.
Okay.
It's figure 2 in the expansions to MGDrivE PRC's paper from 2023. And it shows the simulation
Oh, cool.
of a self-eliminating gene drive.
Okay.
And you can actually see it spreading through the population really rapidly.
Mm-hmm.
And then all of a sudden it's triggered to disappear.
Wow.
And then the original genes come back.
That's amazing.
It's really powerful visual to show, how we're actually
Right.
gaining control over these genetic modifications.
Mm-hmm. It's like we're getting better at it.
Yeah, and none of this would be possible without MGDrivE E2.
Right.
It's like the platform that allows us to model and test all these complex scenarios before actually releasing anything into the real world.
For sure.
And one thing that I think people often overlook is just how much MGDrivE E2 has improved the user experience for researchers.
Okay.
So it's now faster, more efficient.
Okay.
It's got better data visualization tools. It's
Oh,
like
yeah.
they took a powerful engine and made it much easier to drive.
I see.
So that's great news because it means that scientists can spend
Yeah.
less time wrestling with the software
Right.
and more time exploring these nuances of gene drive technology.
For sure.
And that brings us to MGDrivE E3,
Okay.
the latest version
Okay.
of this incredible software.
What are some of the...
What are the key advancements that make MGDrivE E3 so groundbreaking?
So MGDrivE 3 takes a major leap forward by tackling a limitation in the earlier versions. Remember how we talked about how MGDrivE 2
Yes.
simulates disease spread?
Mm-hmm.
MGDrivE 3
Mm-hmm.
actually integrates
Okay.
highly sophisticated epidemiological models.
So it's not just about mosquitoes anymore.
Right.
It's about how the disease behaves within the human population.
Exactly.
Okay, that sounds like a huge step
It is.
towards understanding the real-world impacts.
Yeah.
What kind of detail are we talking about with these new models?
Well, we're talking about things like age structure in the human population, different levels of immunity, and even the effects of like existing interventions
Like bed nets.
like bed nets, anti-malarial drugs.
Malarial drugs.
Yeah, all those factors influence how a disease spreads.
Right.
And MGDrivE E3 can account for all of them
Wow.
in really incredible detail.
That's amazing.
It is.
So MGDrivE E3 is giving us a much clearer picture
Yeah.
of this complex interplay between mosquitoes, humans, and disease.
Yes.
And on top of that, MGDrivE E3 also introduces
Yeah.
a surveillance module.
Really.
So tell us a bit more about that.
So imagine you release a gene drive
Okay.
and you want to track its spread
Mm-hmm.
across a region.
Okay.
MGDrivE E3 lets you do that virtually.
Oh, cool.
So it simulates a network of mosquito traps,
Okay.
and it tracks which mosquitoes are captured in those traps over time.
So you can see the ones that are carrying the gene drive.
Exactly.
Oh, wow. So it's like having a bird's eye view
Yes.
of the gene drive in action.
Yeah.
Watching it spread and seeing how effective it is
Right.
from different locations.
Exactly.
That sounds like a powerful tool
But
in
it's-
monitoring, not just the intended effects,
Right.
but also any surprises.
Yeah, anything that pops up.
You know, things that we didn't anticipate.
For sure.
It's like piecing together a puzzle.
Okay.
Where each trapped mosquito provides a piece of information, and eventually you get this really detailed map of how the gene drive is moving through the population.
Precisely.
That's amazing.
It is.
And there's a figure.
Okay.
Figure 1 in the MGDrivE E3 paper that beautifully illustrates how all of this works.
Cool.
It shows how the mosquito model and the epidemiological model communicate.
Wow.
Creating this incredibly detailed and dynamic simulation.
That's really cool.
So we've got this incredible software that can simulate these complex gene drive scenarios and track their spread.
Okay.
But what does it look like in action?
Yeah, good question.
Did they actually use MGDrivE E3
They did.
to simulate a real-world scenario?
They did.
Okay, what did they do?
So they modeled a CRISPR-based homing gene drive that was designed to block malaria transmission.
Oh, wow.
In a setting similar to the island of São Tomé.
Now, this is a population replacement gene drive.
It is.
It's not about wiping out mosquitoes completely.
Wow.
It's about changing them so they can't transmit malaria.
Exactly.
And the gene drive that they modeled
Yeah.
actually carries 2 genes
Wow.
that make mosquitoes incapable of spreading the parasite that causes malaria.
That's cool.
And the results are really impressive.
Yeah.
The gene drive spreads super fast through the population, and you see a huge drop
Wow.
in both new malaria cases
Okay.
and the overall prevalence of the disease.
So in the virtual world, we're seeing that these gene drives could have a major impact on malaria transmission.
Yeah, it's really encouraging.
Yeah.
And did they also use the surveillance module
Yes, it did.
to see how the gene drive spread across the island?
They used a feature called MGDrivE
Okay.
to place virtual traps across the island
Gotcha.
to track the gene drive's progress.
Cool.
And the simulation output shows which traps captured modified mosquitoes and when,
Okay.
which gives us a really clear picture of the spatial dynamic.
It's like we're watching this thing spread
We are.
across the island.
Yeah,
That's really neat.
it is.
So it's like piecing together a puzzle,
Mm-hmm.
but each trapped mosquito gives you a piece of information, and eventually you get this map of how the gene drive is moving.
Precisely.
Wow, that's the power of MGDrivE E3.
It is.
It lets us play out all these scenarios,
Yeah.
see how the gene drive interacts with the environment and the population, and then ultimately make more informed decisions about whether and how to use this technology.
It's like a really valuable tool.
Yeah. Wow. We've covered so much ground already.
We have.
From the basics of gene drives to these really complex simulations.
Right.
It's clear this technology could be a game changer in disease control.
For sure.
But I think there's a lot more to consider.
Yeah, what else?
Especially when it comes to safety, ethics, and the potential for unintended consequences.
Yeah, we should definitely talk about that.
We'll have to dig into all of that
For sure.
in part 2 of our deep dive.
Awesome. I'm looking forward to it.
So stay tuned, folks.
Yeah, stay tuned.
We'll be back with more insights, thought-provoking questions, and maybe even a few surprises along the way.
Awesome.
Yeah.
I like surprises. Welcome back to our deep dive.
Okay, so last time we were talking about MGDrivE E3 and how it can simulate the spread of a gene drive and even track it using those virtual mosquito traps.
Exactly.
But with all this talk about, changing entire populations, I think it's time we address the elephant in the room.
Yeah.
What about the potential risks?
Right.
What about unintended consequences?
For sure, that's a critical part of the conversation.
Yeah.
One of the biggest concerns with gene drive technology is the possibility of things going off script.
What if it spreads to the wrong population?
Yeah. What if the gene drive spreads beyond the target?
Right.
Or we discover some unforeseen impact on the environment down the line?
Yeah.
Those are big concerns.
So how are researchers trying to address this?
Well, that's where this concept of biodegradable gene drives comes in.
Okay.
Remember those self-eliminating mechanisms we talked about in part 1? Yeah.
That's basically what they're designed to do.
Okay.
Make the gene drive disappear after a certain time.
Gotcha. So like a built-in safety switch.
Exactly.
Yeah. So we could program it to just fade out over time.
Yeah.
Limiting the impact and preventing it from, becoming a permanent fixture.
It's like a round trip instead of a one-way ticket.
I like that analogy.
Yeah.
But what about evolution?
Mm-hmm.
Like mosquitoes are always evolving.
Yeah.
Couldn't they develop resistance to the gene drive?
Yeah, that's a good point.
And wouldn't that just make it ineffective?
That's definitely a valid concern.
Yeah.
And it's something researchers are studying very closely. We need to be prepared for that possibility.
So it's kind of like an arms race.
Yeah.
Where we're trying to outsmart evolution.
In a way, yes.
But evolution's always adapting to overcome our strategy.
It's this constant back and forth between our interventions and the forces of evolution.
Yeah.
But luckily, we've got a few tools to help us with that. Okay, like what?
Well, one strategy is to include multiple guide RNAs in the gene drive design.
Okay, multiple guide RNAs. Can you explain that in simpler terms?
Sure. It's like having multiple keys to the same lock.
Gotcha.
So if a mosquito develops resistance to one guide RNA, there are backups ready to take over.
So it's like a plan B.
Exactly.
Maybe even a plan C.
Yeah, built right in.
That's smart.
Yeah, so we make it much harder for them to evolve complete resistance.
Are there any other strategies?
Yeah, another approach is to target essential genes.
Essential genes?
Meaning genes that are critical for the mosquito's survival.
Gotcha.
So if we can target those, it makes it much more difficult for resistance to evolve.
Because it would kill them.
Without seriously impacting their ability to survive and reproduce.
So we're going after, like, their Achilles heel?
Basically, yeah.
Okay, but this isn't just about science. We're talking about altering wild populations, which raises a lot of ethical questions.
Yeah, for sure.
About our role in manipulating nature.
Absolutely.
Yeah.
You've hit on a really crucial point.
Yeah.
The ethics of gene drive technology are incredibly complex.
It's not simple.
No, we're talking about changing the course of evolution.
Wow.
For an entire species.
Yeah.
That's not something to take lightly.
No, it's a big deal.
We need to carefully consider the potential benefits.
Like saving lives.
Exactly. Saving lives.
Yeah.
Reducing suffering.
Right.
Against the potential risks.
Right. And it's not a decision that scientists should be making on their own.
No, it needs to be a much broader conversation.
Yeah, involving everyone.
Involving ethicists, policymakers.
The communities.
The communities that would be affected.
Yeah.
We need open and transparent dialogue.
Absolutely.
And public trust is essential.
Right.
And that can only come through open communication.
And collaboration.
And collaboration.
So responsible innovation is really the key here.
I agree.
We need to be careful.
Yeah.
And we need to be constantly evaluating the risks and benefits.
As we move forward.
Yeah. So how does MGDrivE fit into all of this?
Well, MGDrivE allows us to test things. And refine our strategies before releasing anything into the wild.
Like a test run.
Exactly.
Yeah.
We can ask what-if questions.
Gotcha.
Explore different scenarios and get a sense of the consequences.
Before it's too late.
Before we make irreversible decisions.
Right. So it's like a safety net giving us a glimpse of the future.
I like that.
And helping us anticipate problems.
Exactly.
But MGDrivE 3,
with
those sophisticated disease models and surveillance capabilities, makes it even more powerful.
It does. We can simulate those ecological interactions, monitor the spread, assess the impact on the target population.
And the environment.
And the broader ecosystem.
Yeah. But even the most advanced model is still just a model.
That's true.
It can't perfectly predict how things will actually play out.
You're right. We have to remember that.
Yeah.
It's a simplification of reality. There will always be surprises.
Unknown.
Things we couldn't have anticipated.
So it's like using a map to navigate a new city.
Yeah.
The map gives you a general sense of where things are, but you still have to be prepared.
For detours.
For those unexpected detours.
Roadblocks.
Yeah, exactly.
Ooh, I never know.
So ongoing monitoring and adaptive management are super important.
I agree. We need to be able to adjust.
Yeah.
Based on what we see.
So we can use MGDrivE to chart that initial course.
Yes.
But we got to be ready to change directions.
Absolutely.
If we need to.
Flexibility is key.
And to make sure MGDrivE is actually effective, we need to feed it the right information.
Garbage in, garbage out, right?
Exactly. So we need good data.
We need good data.
Accurate information.
On mosquito biology, disease dynamics.
Yeah, all of it.
The ecological interactions.
So robust field research is essential.
Yes, we need scientists out there.
Yeah, gathering data.
To improve the models.
So MGDrivE is constantly evolving.
It is getting better.
Getting more sophisticated.
Yeah. It's a great example of how technology and research are working together.
I agree.
To solve these really challenging problems.
For sure.
Okay, so we've covered a lot in this part.
We have biodegradable gene drives.
Yeah, evolutionary resistance.
Ethical considerations.
Data quality.
It's a lot.
We've really highlighted how important MGDrivE is for responsible innovation.
But it's a tool, not a crystal ball.
Right.
We can't just rely on it completely.
Exactly. We still need careful planning.
Yeah.
We need ongoing monitoring.
And communication.
Yeah, communication with everyone involved.
Absolutely.
Okay, so now we're ready to shift gears a little bit.
Okay.
And explore some of the broader applications of gene drive technology.
I'm excited for this. Yeah, because we've been talking a lot about mosquitoes.
Right.
But gene drives can be used for so much more.
Exactly.
So in part 3, we're going to move beyond mosquito control.
Okay.
And look at how gene drives can help us address other problems like invasive species, agricultural pests, and even conservation efforts.
Oh, cool.
So stay tuned, folks.
Yes, stay tuned.
As we delve into the expanding world of gene drives and explore all the opportunities
and challenges
that lie ahead. All right, so we're back for the final part of our deep dive.
Okay.
And up until now, we've been focusing on, mosquitoes and disease.
Right.
But the potential for gene drives goes way beyond that.
It does.
It's pretty mind-blowing.
It really is.
So we're talking invasive species, agriculture, even conservation.
Like a whole range of applications.
Like we're talking about reshaping entire ecosystems here.
Potentially, yeah.
So give us some concrete examples. Like what could these broader applications look like in the real world?
So let's say you have an island.
Okay.
Where there are these invasive rodents
that
are just destroying the native bird populations.
Yeah.
You could use gene drives to control those rodents.
Oh, wow.
And give the birds a chance to recover.
That's amazing.
Or think about agricultural pests.
Okay.
That damage crops.
Yeah.
And force farmers to use all these pesticides.
Right.
Gene drives could offer a more sustainable solution.
Yeah, a more targeted approach.
Exactly, instead of just blanketing everything with chemicals.
Yeah, it's like we're moving from, these blunt instruments to precision tools.
Exactly.
But I imagine there are some challenges.
There are definitely challenges.
That come with applying this technology in such different ways.
Especially when we move beyond mosquitoes.
Right.
The ecological considerations get a lot more complex.
So how do we make sure we're using this technology responsibly?
Well, MGDrivE comes in handy here again. But this time, we're not just simulating mosquitoes.
Right.
We're building virtual models of whole ecosystems.
Oh, wow.
To understand the potential impacts.
On all the different species.
Right, all the interactions.
And how they're all connected.
Yeah, it's like ecological modeling.
Trying to predict how these changes might ripple through the whole environment.
Exactly. And these models help us identify potential risks.
Okay.
Before we actually do anything.
Give me an example.
Okay, so what if a gene drive meant for an invasive plant?
Okay.
Accidentally spreads to a native plant.
Oh, no.
Yeah, that would be bad.
Yeah.
MGDrivE can help us catch those kinds of things.
And then design safeguards.
Exactly. It's like playing chess.
Okay.
Thinking several moves ahead.
Trying to anticipate the consequences?
Yes, and that leads to the idea of stage testing.
Okay.
We wouldn't just release a gene drive into a complex ecosystem.
Right, start small.
Right, we start with controlled lab experiments.
Okay.
Then maybe move to isolated field trials. Gathering data every step of the way.
So really carefully monitoring things.
Exactly, and only after all that would we even consider broader implementation.
So it's a very cautious approach.
It is.
Which is reassuring.
Yeah.
But even with all these precautions, there's still going to be unknowns.
It's true. We can't predict everything.
So we need to be flexible.
Adaptive.
Ready to change course if necessary.
It's like launching a ship.
Okay.
But keeping your hand on the rudder.
Ready to steer.
Yeah, in case you need to change direction.
So we use MGDrivE to get us started, but then we have to be ready to adapt
to whatever happens
to the real world.
Yeah.
And this is where you, the listener, come in.
Yeah.
Because as gene drives, expand into all these different areas, it's even more important to stay informed.
And engage in these conversations.
What do you think about all of this?
Yeah, what are your thoughts?
What boundaries do we need?
What opportunities are there?
Because these aren't just scientific questions anymore.
These are societal questions.
And the future of this technology isn't set in stone.
It's up to us.
Yeah. We get to decide how we want to use it.
Through informed choices.
Informed by science.
By ethics.
And what we want the world to look like.
Exactly.
It's amazing that we all get to play a role in shaping this.
I agree. It's a really exciting time.
So as we wrap up our deep dive,
yeah,
I'm just amazed at the potential of gene drives.
Me too.
To address these huge challenges.
Like it could really change the world.
But it comes with a lot of responsibility.
Yeah, we have to be careful.
We need to be thoughtful.
Proceed with caution.
Yeah, guided by science.
and ethics.
And a respect for, the interconnectedness of everything.
It's all connected.
MGDrivE is a great tool.
It is.
To help us navigate all of this.
But it's up to us to use it wisely. So one final question to leave you with.
Okay.
What role do you envision for MGDrivE technology in shaping the future? That's a good one to think about.
It's a big question.
It is.
But it's one that's worth asking.
For sure.
As we enter this new era.
This new era of genetic possibilities.
That's it for this deep dive.
Thanks for listening, everyone.
We'll see you next time.